Good morning, my dear students. I welcome you all to my next online class, chapter 12, plant anatomy and plant physiology. So far we have finished 2 classes in this chapter, in which we have learned this just intro. We have learned only the introduction of this lesson, where we learned about the various different tissue systems like epidermal tissue system, ground tissue system and vascular tissue system. Then we have learned the various different types of tissues of vascular categorized based on their arrangement. We have also learned another important concept, that is the difference between a dicotyledon and a monocotyledon. And in today's class we are going to learn about the anatomy of dicot root, monocot root and dicot stem, monocot stem. As we discussed in previous class, the ground plan, what you are looking at is the ground plan of transverse section of dicot root on the left hand side, and the ground plan of transverse section of monocot root on the right hand side. First, you be clear with this concept, that is in case of dicot root, where the vascular bundle will be present at the center without the presence of pith. That is, in case of dicot root, the pith will be found only in young roots. When the roots mature, it results in the absence of pith. So that is, in some charts or in some diagrams, you can find the pith at the center. So inside the vascular bundle you can find the pith. But here, if it is a mature root, you don't have pith at the center. But at the same time, with regard to a monocot root, where you come across some tissues at the center, which is actually known as pith. That makes the first difference between a dicot root and a monocot root. Similarly, you have this vascular tissue. The vascular tissue is radial in nature, both in case of dicot root and monocot root. Okay, so radial, that is, the xylem and phloem are not at a particular radius from the periphery or from the epidermis or endodermis. That is actually known as radial. So, which is common for both dicot root and monocot root. Similarly, you can come across the vascular bundle exarch in nature. I hope you know what is meant by exarch. That is, metaxylem at the center, which will be bigger in size, and protoxylem towards the periphery. This is actually known as exarch. So this is the main common characteristics of dicot root and monocot root. Again I repeat, one, you come across pith only in young dicot roots, but not found in adult or matured dicot roots. But in monocot roots, you have a pith at the center of the vascular bundle. Second is, common feature is they are radial in nature. The vascular arrangement, the arrangement of vascular tissue is radial in nature, and they are exarch. That is, metaxylem at the center and protoxylem towards the periphery. Okay. In dicot root, we have this 4 important tissues. One, the outer epidermis, which covers and protects the whole structure. And next, you have inside cortex tissues, so which is actually a ground tissue. Then third, we have another circle inside which vascular bundles are found, and this is actually known as endodermis. Epidermis, endodermis. In between epidermis and endodermis, you have a layer of cortex tissues, and finally the vascular bundle, that is, it is known as actually stele, S-T-E-L-E, stele. This stele comprises of three important tissue systems, namely pericycle, vascular bundle, and pith. So this is the overall idea about Dicot root. Again I repeat, outer epidermis, then next to the epidermis you have cortex tissues, then next to the cortex you have endodermis in the form of a circle, okay, in the form of a covering, and inside that you have stele, and the stele comprises of pericycle, vascular bundle, and pith. Right, now let's get into the structure of the Dicot root. So this is a transversal section of a Dicot root, which comprises of four important layers. One, epiblemma or rhizodermis or piliferous layer. That is, the epidermis of Dicot roots are actually known as epiblemma or rhizodermis or piliferous layer. Okay. Then second is you have this cortex tissues, which is next to this epidermis. Okay. And the third one is you have endodermis. This is the third layer. And finally you have the vascular bundle, which collectively known as stele, S-T-E-L-E. Okay. So these are the four layers of Dicot root. Now let's start with the epidermis, which is also known as epiblemma or rhizodermis or piliferous layer. So first point, epidermis is the outermost layer of Dicot roots. So it is the outermost layer of all the plant parts, and here it is the outermost layer of plant Dicot roots. Second, in roots, either it is Dicot or Monocot, you don't have the presence of cuticle as well as stomata. So you know stomata, which is a minute pores that are found in the leaves, abundantly found in the leaves, less found in stem, and also less number or absent in xerophytes. So you know stomata through which respiration and transpiration takes place. Okay. And now we are learning another point, that is cuticle. What is cuticle? It is a thin layer which is found covering the epidermis of stem especially. Okay. So this cuticle is absent in roots and absent in Dicot root. Okay. So again I repeat, the epiblemma or rhizodermis or piliferous, which is actually the epidermis, is the outermost layer. Cuticle is absent, stomata is absent. Okay. And here you come across small hair-like structures, and those are known as root hairs. Okay. So in root you have root hairs, and the specialty of root hairs is they are mainly involved in the absorption of water and minerals. The function of root hair is absorption of water and minerals. I hope you know that root absorb water and minerals, and actually even though you have primary root, second root, and tertiary root, it is only the root hair which absorbs or which is involved in the absorption of water and minerals. Okay. And one more thing, it's not that water absorbed separately and minerals absorbed separately. So it is very difficult to absorb minerals. It can be adsorbed but not absorbed. I hope you understand. Okay. So minerals is a solute, so it can only be adsorbed, not absorbed. So mineral, they mix with water to form mineral solution. Okay. So that is like how you have salt plus water is equal to salt solution. Sugar in water is called as sugar solution. If you dissolve salt in water, if you dissolve sugar in water, so they are known as a salt solution or sugar solution. Like that you have minerals dissolved in water inside the soil, and that is actually known as mineral solution. So it is very easy. Okay, you don't need to confuse. Minerals like sodium, potassium, calcium, magnesium, manganese, iron, etc., which are found abundant in soil based on the fertility of the soil. Okay, and when water is there in the soil, it mixes with the water, and the root hairs absorb those minerals in the form of mineral solution, which is done only by root hairs. Okay, so this is about your epidermis. Okay, outermost layer, cuticle and stomata absent, unicellular root hairs are present. The root hairs are unicellular in nature. Then the second layer is cortex. So this fully, actually if you look into a ground plan, I showed properly. So the whole cells are known as cortex. They are multi-layered, that is, they are found abundantly, one above the other or one below the other. Okay, and it is the largest zone compared to epidermis, compared to endodermis, compared to vascular bundle. It is the largest zone and made of thin walled parenchyma. So each and every cell is actually a parenchymatous cell. So most of the cases, when you learn cortex, these cortexes are made of parenchymatous cells here with intercellular spaces. Okay, so I hope you understand. One, it is multi-layered, so there are many layers of cells. So it is a 2D diagram, so you cannot understand that. But if you look into 3D, if you cut a root, you can understand the cells are placed in several layers, one above the other. And this is the largest zone. And third point is, it is made of each cell is made of thin walled parenchymatous cells. So that is, we have learned before, collenchyma, parenchyma, sclerenchyma. And these cells are parenchymatous in nature with intercellular space. So you will learn later in some cortex cells there will not be space. Can you understand here? So in between two cells you have a space. In between the cells you have space. This space is actually known as intercellular space. Inter means in between. In between other cells you have space. That is known as intercellular space. And the main function of cortex is it stores food and water. So that is the main function of parenchyma cells. Since they are parenchymatous in nature, they have the ability of storing food and water. Okay, that is about cortex. Then third is endodermis, which is the innermost layer of cortex. Okay, first epidermis, then cortex, and third one is endodermis. Okay, the cells are barrel shaped. I hope you know barrel. In those days, they store water or wine or liquor in barrels. In that shape, these cells are found. So that is actually known as barrel shaped. Okay, so the cells are barrel shaped, and here, unlike your cortex, you can find the cells are closely packed. There is no space. Okay, that is actually known as closely packed. Okay, so they are the innermost layer of cortex. Okay, epidermis, cortex, and innermost is endodermis. They are barrel shaped and they are closely packed, no intercellular spaces. The next is, here you come across the presence of Casparian strips. These Casparian strips, they help in the movement of water and dissolved salts from the cortex into the xylem. So this is actually this is xylem, like a cross, it is present at the center. Okay, so from here, that is actually root hairs absorb water. From root hair it reaches epidermis, from epidermis it reaches cortex, and from cortex it should reach the xylem, such that the water is transported to other parts of the body. Okay, so xylem is actually involved in the transportation of water and minerals, and it should enter, so it should pass this endodermis. You have a barrier. Actually, endodermis is a barrier which does not allow everything inside, because something important is inside. Okay, that is xylem and phloem, important. So the water that is found in the cortex should enter into this vascular bundle, and it is only through this Casparian strips. The next is stele. Whatever that is found inside the endodermis is actually known as stele, which includes one, pericycle, second, vascular bundle, and pith. As we discussed in the beginning, in roots, in dicot roots, only in young roots you come across pith at the center. So if it is matured, there is no pith at the center. So you can find only xylem. Okay, so which means this is a matured root. Okay, Then second is with related to pericycle. So pericycle is a single layer and a thin walled and which is an origin of lateral roots. That is, from this pericycle, okay, so from this pericycle where some roots arises rarely, okay, some lateral roots arises, that is the significance of pericycle. Okay, one pith which is not found in matured roots, which is found only in young roots. So here there is no pith. Second is pericycle, which is a single layered structure, okay, and the second is in pericycle this results in the origin of lateral roots, that is from here certain roots arises. Then third, with related to vascular bundle, that is xylem and phloem. So this is phloem and this one is xylem. So when you look into a diagram, you come across colors, each one will be given in a color, and this is just to differentiate or make you easy to understand. But actually if you look into a root or a stem or any part of a plant, it is not colored actually, except your chlorophyll, chloroplast, which is greenish in color, or other pigments which gives other colors in fruits. You don't come across colors inside the tissues. If at all a slide is prepared, what they do is that they add dye. So that dye gives a color for the cells inside, and thereby you come across mostly single colored. The slides will be single colored, but that color is not actually the color of the plant part, but it is the color of the dye that is added. Similarly here in diagrams or photos, especially where you come across colors given for each and every structure, and it is mainly to differentiate the parts and to make you easy to understand. Okay, right. So with regard to vascular bundle, here in dicot roots, the vascular bundle are radial in nature. They are exarch and tetrarch. Okay, so exarch we have learned before, that is where you come across metaxylem at the center and protoxylem towards the periphery. This condition is known as exarch. Okay, in the middle larger, that is actually metaxylem. Okay, then second is in between you come across certain conjunctive tissues. Okay, so where you come across certain conjunctive tissues, and those tissues are made of parenchyma cells. Pith is absent, but you have some certain tissues, and they are made of parenchyma cells. The next what we are going to learn is monocot root. So first let us finish with the ground plan, then we'll go to the full structure. With related to this ground plan, which describes the four important structures what you have learned in dicot root. That is, first one is the presence of the outermost epidermis. So this epidermis, which also known as epiblema or rhizodermis, like your dicot root. In monocot root also, the outer epidermis is also known as epiblema or rhizodermis. Then next to this epidermis, the outermost line is epidermis. Next to this epidermis, you have this cortex layer, which comprises of again parenchymatous cells. So here also, since it is parenchymatous cells, the cortex cells they are involved in the storage of food and water. Okay. Then third layer is endodermis, the circle which is actually known as endodermis, and inside this endodermis you have xylem and phloem. Okay, so you have xylem and phloem tissues, and at the center you have pith. So almost similar. Only difference is there in dicot root, in matured roots there is no pith, but here you have pith at the center. That is the main difference between dicot root and monocot root. So with this four topics, we'll enter into the concept. Okay, do you want me to repeat? One, epidermis. Second, cortex. Third, endodermis. And fourth one is stele, which comprises of pericycle, vascular bundle and pith. So this is the transversal section of a monocot root. Like a dicot root, almost the parts are similar. Okay, so that is first, presence of epidermis. This epidermis is also known as rhizodermis or epiblema, or it is also known as piliferous layer. Okay, the outermost structure, epidermis. And absence of cuticle and stomata. And third one is they are unicellular. They have unicellular root hairs. Okay, so can you find this root hairs which are unicellular in nature? The main function of epidermis is this covers and protects the whole system. Okay, epidermis which covers the full tissue, all the tissues inside, and it protects. Second one is root hairs which are involved in the absorption of water and minerals. Okay, the second layer is cortex cells. So these cortex cells, as we discussed in dicot root, these also are parenchymatous in nature. They are made of parenchymatous cells with intercellular spaces, and the main function of this is to store food and water. Okay, epidermis protects and covers the cell. Root hairs helps in the absorption of water and minerals. Cortex cells, since they are parenchymatous in nature, they store food and water. Then next is endodermis. This is in the form of a covering. Okay, so a single layered structure called as endodermis. The main function of endodermis is it covers and protects the vascular bundle. And as we learned in dicot root, they are also barrel shaped, closely packed, and inside this endodermis where you have stele. Stele is the collection of three important tissues. One, vascular bundle, that is xylem and phloem. Second is the pith, and third one is a single layer of pericycle. Okay, so let me start with the pericycle first. So pericycle is single layered. Okay, so it is not made up of many layers like this. So this is actually known as multi-layered. If you look at epidermis or endodermis or pericycle, they are single layered. Okay, so they are single layered. Pericycle is single layered, and it involves in the origin of lateral roots. That is, from here certain lateral roots arises. That is the use of pericycle. Then next is pith. Okay, which is at the center. They are also made up of parenchymatous cells, and because of that you come across abundant storage of starch, that is food, in this pith. The final is xylem and phloem. So you come across xylem and phloem at different radii. Hence they are known as radial arrangement, since they are at different radii. See, this is a circle, and from here at one radii you come across phloem, at another radii you come across xylem. So hence they are known as radial arrangement. The arrangement of xylem and phloem is known as radial arrangement. And the next is they are exarch in nature. The vascular bundle is exarch in nature. That is, when you look into the xylem tissues, metaxylem towards the center and protoxylem towards the periphery. Periphery means outside. Okay, so since protoxylem at the periphery and metaxylem at the center, this nature is actually known as exarch. So this is about your monocot root. Epidermis, cortex, endodermis. Inside you have stele. The stele comprises of a single layer of pericycle, presence of pith at the center, xylem and phloem. The vascular bundles, they are radial in nature, and second one is they are exarch. Metaxylem at the center and the protoxylem towards the periphery. The next is dicot stem. So this is the ground plan of a dicot stem. So from this what you learn is, like in root you have external epidermis, and next to epidermis you have cortex cells. Okay, a small layer of cortex cells, and that cortex cells are divided into three, which we will learn later. Okay, three layers are there in cortex cells, which we will learn later. Then the third layer is endodermis, blue circle. Okay, blue color line, that is actually known as endodermis. And finally, like in root, you have inside stele. Stele, which comprises of pericycle, pith and vascular bundles. Another important importance that is found in dicot stem is the vascular bundles are arranged in the form of a circle, which we discussed in even in last class. Okay, So that is an important classification or differentiation between a dicot stem and a monocot stem. Okay, in monocot stem we are going to learn where the vascular bundles are scattered in the cortex, unlike here. Okay, again I repeat, one outermost epidermis. Okay, so just have an idea, an external idea, outer overlook. Okay, so first you have outer epidermis. The next to epidermis you have cortex cells which are divided into 3 layers. Then next is endodermis. Inside this endodermis where you have the stele comprising of pericycle, pith and vascular bundle. Okay, now we'll discuss about the enlarged structure of this dicot stem. Okay, so where in all the enlarged structure you come across a part of this overall structure or overall ground plan. Okay, a part is actually discussed, either it is dicot root or monocot root or dicot stem or monocot stem. So everywhere a part is enlarged, and that is what I am discussing. Okay, yes, this is the section enlarged of a transversal section of dicot stem. So in this, first let us start with the outermost structure, that is epidermis. Okay, so the epidermis, so actually don't worry about the internal structures. When you start learning these internal structures, then it will be easy for you. Okay, so if you are, first of all you be thorough with the ground plan, and then you can start concentrating on this structure. Okay, don't worry, it appears like complicated. When I start teaching and when I finish this dicot stem and monocot stem, just have a comparative idea of both the structures, then it will be easy. Okay, never worry about this structure. Okay, first let us start with the epidermis. Okay, so here in case of dicot stem, okay, so now you have to compare the root with the stem. So far we have learned about root, where cuticle is absent there, stomata is absent there, and the presence of root hairs there. But here presence of stomata, presence of cuticles, and next is here you have minute stem hair. So that makes the difference between a root and a stem. Okay, so let me start with epidermis. First of all, as usual, it is the outermost layer. Like in root, even in stem, epidermis is the outermost layer, and it is made of a single layer of parenchyma cells. Okay, so like root also, it is made up of single layer of parenchyma cells. Here also you come across single layer of parenchyma cells. Then the outer wall is covered with cuticle. So can you find a line here, a small line which covers the epidermis? That is actually known as cuticle. That small line is actually known as cuticle, which is found mainly in stem. In some leaves it may be present, but in stems you have this cuticle. The speciality of cuticle is in some cases it avoids transpiration, and in some cases it enhances transpiration. So based on the plant, based on their habitat, the function of cuticle differs. Okay, and you have small hairs. You might have seen in some tender plants where the stem comprises of minute small hairs. If you have not found, kindly go and observe. And those hairs, since they are found in stem, they are known as stem hairs. Root hairs are found in roots, stem hairs are found in stem. Okay, then the second layer is cortex. The cortex is made up of 3 regions, which includes hypodermis, which comprises of or made of collenchyma cells, and the second layer is middle cortex, which is made up of chlorenchyma cells. Okay, this is collenchyma. The second layer is chlorenchyma, and the third layer which is actually known as inner cortex, and it is made up of parenchyma cells. So these 3 layers comprises of cortex. Okay, and next to that you have endodermis. So that you should not consider. Maybe here it is wrongly marked, so that should not be considered. So only 3 layers, that is hypodermis, middle cortex and inner cortex. Middle cortex, inner cortex. Outer cortex, that is hypodermis, which is made of collenchyma cells. The middle cortex is made up of chlorenchyma cells, and the inner cortex, the third region, is made up of parenchyma cells. Okay, so here this collenchyma, which actually gives a mechanical support to the stem. epidermis, which covers and protects all the structure. This hypodermis, which is made of collenchyma cells, it gives a mechanical support. The middle cortex, since they are chlorenchyma, they are known as chlorenchyma because they comprise of chlorophyll tissues or chloroplasts. They are involved in photosynthesis. They are green in color and they are involved in photosynthesis. This is mainly found in young stems. So you come across tender plants, young plants which are greenish in color. The stem will be greenish in color, which comprises of chlorenchyma, and so they are involved in photosynthesis. And the third layer is parenchyma cells, that is inner cortex, which is involved in the gaseous exchange as well as in food storage. Whenever when you come across parenchyma, as I said before, it is involved in food storage. And here also this is involved in food storage. At the same time, it is also, to some extent, it is also involved in the gaseous exchange. Okay. The next layer is endodermis. Okay, the blue color structure is actually known as endodermis, which is the innermost layer of cortex, that is next to cortex. That is why it is known as innermost layer of cortex. And it consists of single layer of barrel shaped cells. Everywhere you come across this word, endodermis, made up of barrel shaped cells, and it contains starch grains, and so they are known as starch sheath. Okay, inside you have many starch stored there, and so sheath means a covering, and so it is known as starch sheath. Endodermis is also known as starch sheath. The third one is your stele, which comprises of one pericycle, second vascular tissues, that is xylem and phloem, and third one is presence of pith. The pericycle is present between the vascular bundle and the endodermis. So in between the endodermis and the vascular. So this is fully called as vascular bundle. So you have a phloem tissues and xylem tissues. Okay. So together they are known as vascular bundle. And in between these two, that is endodermis and the vascular bundle, you have the presence of parenchymatous cells alternating with sclerenchymatous cells. So that is actually known as pericycle. Okay. So you have sclerenchyma and parenchymatous cells. Together you call it as pericycle. Then next let me finish with the pith, because they are large. I mean, this comprises of many large cells with more intercellular space. Can you find intercellular spaces? Okay. So intercellular, that is in between the cells you have spaces, and since they are parenchymatous in nature, as usual, pith is parenchymatous in nature, and so they are involved in food storage. Okay. Then coming to vascular bundle. So here in dicot stem, the vascular bundle is conjoint, collateral, endarched and open. Okay. So it is conjoint in nature. Okay. So that is, it is not radial. That is, you come across this vascular bundle at the same radii. Okay, so you come across phloem here and you come across xylem. So since they are at a particular radius, if you compare this dicot root, where you will find phloem in one corner, xylem in another corner. But here it is not so. The vascular bundles, they are at the same radii from the periphery, either from epidermis or from the endodermis. If you calculate, they are from at the same radius, and so they are known as conjoint. And second is they are collateral. What is meant by collateral? So here you come across the vascular bundle which are conjoint in nature. Conjoint, that is where the xylem and phloem are from a particular radius. That is, if you look into a ground plan, you can understand they are in the form of a circle. So surely they are at a particular radius from the periphery. Whereas in the root where you come across a radial, they are not at the same radius. Okay, so that makes the difference between a radial and a conjoint. Okay, first one is conjoint, they are conjoint in nature, and second one is they are collateral in nature. Okay, so you have this collateral and bicollateral. So if you come across a xylem at one particular radius, and above and below if you find the phloem, that is actually known as bicollateral. But here you don't come across two phloem, you have only one layer of phloem, and you have a layer of xylem, maybe protoxylem and metaxylem, but put together you have here xylem and here phloem, that's all. So that makes the difference between collateral and bicollateral. Again I repeat, if you have xylem, above and below if you have phloem, that is known as bicollateral, but here it is not so. You have a layer of xylem and a layer of phloem, and so they are known as collateral in nature. Then third one is here end arch in nature. So in roots we found bigger cells at the center, smaller cells at the periphery. That is metaxylem here, protoxylem here. But here in stem we come across protoxylem towards the center and metaxylem towards the periphery. That is the external structure, external outwards. Okay, so if metaxylem is towards the periphery and protoxylem towards the center, that is actually known as end arch. So kindly understand the difference between exarch and end arch. Okay, so roots, they are exarch in nature, and stem, they are end arch in nature. And finally open, okay, conjoined, collateral, end arch and open. What is open? As we learned before, the presence of cambium tissues. If there is presence of cambium tissues, that results in the secondary growth of other tissues. That is, it initiates the growth of other tissues, and hence it is known as open type of vascular bundle. That is, it helps in the increase in number of the tissues, and so this kind of vascular bundle is known as open type. That is, if cambium is present, it is known as open type. If cambium is absent, then it is known as closed type, because the absence of cambium. So if there is no cambium, there is no external growth, which means it is closed. The vascular bundle is closed. But here, presence of cambium, it initiates the growth, it initiates the size, etc., and that's why it is known as open type. I hope you understand. Okay. And finally, one importance of this dicot stem is with regard to this phloem. I hope you know there are 4 types of phloem, which includes sieve tubes or sieve elements, then companion cells, phloem parenchyma and phloem fiber. In case of dicot stem, absence of phloem fiber and phloem parenchyma. Phloem fiber and phloem parenchyma are absent. So what is present is phloem comprises of only 2 types of phloem tissues, that is, one sieve elements or sieve tubes, and second one is presence of companion cells. Absence, again I repeat, absence of phloem fiber and the phloem parenchyma. This is another importance of dicot stem. Today's assignment is, in monocot stem, if you look into a ground plane of monocot stem, where you come across outer epidermis, then secondly you have another layer of hypodermis, which is made of sclerenchymatous cells, and inside you come across cortex cells, and thirdly, finally, you have vascular bundles. Okay, so these are the basic 4 structures of monocot stem. One, like in all other plants, you have externally outer layer known as epidermis. Then next to epidermis, you have a layer of hypodermis, so which is found immediately next to epidermis, which is made up of sclerenchyma cells. And third one is you have ground tissues, the ground tissues which are none other than the cortex cells. Then finally you come across vascular bundles. The main difference between a dicot stem and a monocot stem is, in dicot stem the vascular bundles are neatly arranged in the form of a circle. Do you remember the diagram which I described just now? It is neatly arranged in the form of a circle, but in monocot stem it is scattered all over the cortex cells. Which is scattered? The vascular bundles are scattered all over the cortex cells or all over the ground tissue. That is the main difference between a dicot stem and a monocot stem. Now let us learn the enlarged structure of a monocot stem. So this is the enlarged structure of a monocot stem. So where you come across outer epidermis. the epidermis. Here in monocot stem, the epidermis lacks hairs. So where in dicot stem we learned presence of stem hair, but in monocot stem there is no stem hairs, absence of stem hairs. And here second, in case of monocot stem, the stomata are less in number compared to dicot stem. Here stomata is present but they are less in number. Next layer is hypodermis. As I said before, it is made up of sclerenchyma cells, and then and there you come across certain chlorenchyma cells also. In hypodermis you come across mainly sclerenchyma, and in between you come across certain chlorenchyma cells. Since they are chlorenchymatous, which means they will be greenish in color, and the second one is they are involved in photosynthesis. Okay. Then basically the function of hypodermis is, as we learned in dicot stem, which gives mechanical support. Okay, this is about hypodermis. Then third is the ground tissues, which are found abundantly inside epidermis and hypodermis. So the ground tissue here, which is not differentiated into, as we learned in dicot stem. Dicot stem, it is differentiated into three layers as endodermis, cortex, pericycle and pith. So it is not like that here. So the ground tissue is found, but it is not differentiated into any other different types. So fully it is covered with ground tissue. Okay. With related to this vascular bundle, okay, so you come across vascular bundle which appears like a skull. The head region of a skeletal system is actually known as skull. So can you find like a two and like your mouth? So hence it is known as skull shaped structure. And as we discussed in the beginning, that is in the ground plan, the vascular bundle is scattered all over the ground tissues. Then the vascular bundles are conjoined. That is, here the vascular bundles they are found at a particular radius. See, each vascular bundle is at a particular radius. That is, we are talking about both xylem and phloem. So xylem and phloem, they are packed in a vascular bundle. So from the periphery it is at a particular radius. Okay, so xylem and phloem at a particular radius. So that is actually known as conjoined. Then second one is collateral. That is, what is collateral? That is, there is no two phloems. So even in last topic I said, if xylem is there, phloem is there, that is known as collateral. If xylem is there on both sides, if you have phloem, that is known as bicollateral. Here you don't have two layers of phloem, so that's why it is known as collateral. Okay, again I repeat, conjoined, second one is collateral, and third one is endarched. I hope you remember what is endarched. That is protoxylem towards the center and metaxylem towards the periphery. So this is the center. From here, which is near, protoxylem is near and metaxylem is far away, that is towards the periphery, and this kind of arrangement is known as endarched. Okay, then last one is absence of cambium, and so it is known as closed type of vascular bundle. Okay, again I repeat, vascular bundle, first of all it is scattered all over the ground tissue. Second one is you come across skull shaped structure. Each vascular bundle is like a skull. And third, they are conjoined. Conjoined means from the periphery, each vascular bundle is at a particular radius, comprising of both xylem and phloem. Since they are packed, you say it is at a particular radius. Next is collateral. Collateral means you have one layer of xylem, one layer of phloem. That's all. When you have two layers of phloem, that is xylem at one position, above and below, if you have phloem, that is actually known as bicollateral. But here it is not so. You have one layer of xylem, one layer of phloem, and so it is known as collateral. The next is endarched. That is protoxylem towards the center, metaxylem towards the periphery, and so it is known as endarched. And finally, absence of cambium, and so it is known as closed type. Okay. Now here, if you look into this structure, each vascular bundle is surrounded by few layers of sclerenchyma cells. Okay, so you come across some sclerenchyma cells, and that condition is known as bundle sheath. Last topic we learned starch sheath. That is covering in your vascular bundle, which comprises of starch, and so it is known as starch sheet. But in monocot stem, here you come across sclerenchyma cells, which forms a vascular bundle, and so it is known as bundle sheet. I hope now you can clearly understand the structure. I said the skull shaped. Okay, so now you can understand the skull shaped structure of vascular bundle, which comprises of xylem and phloem. So this pink color structures are known as xylem, and here you have the structure of phloem, xylem and phloem. Okay, so if you look into a vascular bundle, so actually a vascular bundle is enlarged here, magnified here. First let me say something about this xylem tissues. Xylem is arranged in the form of V or sometimes Y-shaped structure. In some vascular bundles you will find another xylem here. So it appears like Y. Here it appears like V. So the xylem is in the form of a V or Y-shaped structure. Okay. In a mature vascular bundle, the lowermost protoxylem. So this is protoxylem, this is metaxylem. So you can say based on their structure, metaxylems will be bigger and protoxylems will be smaller. If you look into this xylems, that is protoxylem and metaxylem, the protoxylem will start to slowly disintegrate and forms a cavity-like structure, which is actually known as protoxylem lacuna. Okay, so the protoxylem disintegrates and forms a cavity. Cavity means empty space, like a vacuole. Maybe in lower classes you have learned inside a cell where you have vacuoles. Like that, cavities are found because of the disintegration or death of protoxylem, and that cavity is known as protoxylem lacuna. So these are all important points. Note down properly, memorize the words. Okay. Then next, with related to phloem. So in phloem, the phloem comprises of sieve tubes or sieve elements and companion cells, as we discussed before. That is in dicot stem, where you have companion cells and sieve tubes, but absence of phloem fiber and phloem parenchyma. Okay, so both in dicot stem and monocot stem you have this common feature, that is presence of companion cells and sieve tubes, which is also known as sieve elements, absence of phloem fiber and phloem parenchyma. So I think with this we'll end today's session. In next period I am going to start with dicot leaf and monocot leaf. So as I said before, keep this in mind, that is first concentrate on the names, on the terms which we have discussed before, that is exarch, endarch, classification of vascular tissue, etc. That introduction. Then only you will understand about the ground plan. So secondly, you go through the ground plans, compare one type of plant part with another type, compare with the dicot with the monocot, that is dicot root with the monocot root, dicot stem with the monocot stem, like that. Then thirdly, you concentrate on the full structure of this plant parts, that is learn thoroughly the dicot root, learn thoroughly the monocot root. So most of the points will be similar, only some characteristics differ from one from other. For example, characteristics of dicot root and monocot root, you will find only minor differences. Again in dicot stem, monocot stem, certain differences. So that should be memorized, so that it will be easy for you when you go to a higher secondary. Okay, so because as I said before, you are going to learn again all these concepts in higher secondary. 11th standard, 12th standard, if you are going to school, okay, so in that case you will be having practicals, and in practicals you have all this transversal section of various different plant parts like dicot root, monocot root, dicot stem, monocot stem, etc. So you should be ready for that. Okay, so now itself you learn, you be ready such that it will be easy for you to learn again the same concept with some more extra points. So surely you will be learning extra points there, concepts there, then it will be easy for you. So if you leave now, it is going to be a very difficult task in your higher secondary, because let me add one more thing, that is when you are in adolescent age, that is from 9, 10, 11, 12, I mean teenage, okay, so that is from 13 years to 19 years, many kinds of disturbances will be there. So you should be aware of that, which are all caused by certain hormones, etc. So already I've learned in 8th standard, reaching the age of adolescence. So you all know about that. So you should overcome those disturbances, those obstacles, and thereby concentrating on music, or concentrating on education or learning, etc. At the same time, I hope you remember what you have learned in 8th standard, reaching the age of adolescence, where in that topic it will be, you might have learned that this teenage is the right age to memorize as many things as possible. Keep that in mind. Okay, so if you live this age, in future it will be difficult for you to memorize or store in your brain. Okay, so keeping this in mind, concentrate, read more. It's not only this subject or that subject or this lesson, that lesson. So you just concentrate on all the topics, all the concepts, all the subjects. Try to memorize as many things as possible, and that too positive things. Okay, right. Anyhow, let me finish today's session with this. Attend the online test perfectly. Try to score more marks, which means you have to go through the book or go through the concepts. Then only you'll be able to attend the online test perfectly and get full marks. Okay, so don't take this lockdown as lethargic. Okay, so there are many who skip the concepts and directly going to assignments or going to the online test. So it should not be the case. Okay, so keep that in mind. Try to be at least sincere to your extent. Okay, so each one have your own capacity to be sincere, to be punctual, to learn more, etc. And you try to go to that extent of your limit. That limit, I don't know. So you only should be analyzing, and based on that you only have to proceed. Okay, so and develop those skills. Okay.